Nyquist. I'm a professor of anesthesia, critical care medicine, neurosurgery, general internal medicine at Johns Hopkins, and I work in the neurocritical care unit there. Today I'm going to talk about biomarkers and brain injury, and then I'm going to talk a little bit about systems biology approach to acute brain injury. And hopefully I'll change your perspective on biomarkers away from the magic indicators of brain injury to something that really grants insight into the biological mechanisms of brain injury. So what is a biomarker? This is actually an important thing here. The definition, here's a definition. And basically, a biological marker refers to a broad subcategory of medical science that's objective, measurable indicators of a medical state observed from outside the patient. They can be measured accurately and reproducibly. And they stand in contrast to medical symptoms, which is really a report of the patient's feelings or experience of disease, and can often be not necessarily untrue, but often difficult to quantify. FDA biomarkers are key right now because they're in this precision medicine mode. They want to demonstrate that they have a capable facilitating development of medical products. They want to spur innovation. And when used in the right context, biomarkers have the potential to help expedite patient access to safe and effective treatments by reducing the time and cost of clinical trials while maintaining patient protections. So back in the Obama administration, there was an initiative for precision medicine. So a large independently funded mandate from Congress outside the NIH, but eventually run by the NIH. And there's a whole bunch of science going on that's been funded by this initiative. So there's all kinds of biomarkers. It can be almost any observed characteristic as long as it grants insight into an underlying disease independently of the symptoms of the disease. So it could be skin color, it could be pupil response. But usually when we think of biomarkers, particularly in the context of neurologic disease, we're really talking about MRIs, CTs, and any subcategories of imaging. So electrophysiology, EEG and EKG are also biomarkers. And then there are genetic markers that sometimes can be biomarkers of disease, but often are not. And it really depends on how strongly the gene is related to the expression of the disease. And then there's serological markers. We're often obsessed by that. And this would include CSF markers. We look for these things as markers of, so quote unquote, indicators what's happening. But really, they're not always all that valuable at indicating the course or the diagnosis. So in neurocritical care, we become very focused on multimodality monitoring. And really, we need to ask ourselves a series of questions. What does it measure? What does what we're measuring mean? Is information valuable? Does it really lead to better prognostication? Can we effectively intervene based on the information? Of all this, I was, a strong proponent, almost 20 years ago when Lycox and tissue oxygen monitor came out. Roz Bullock, who was the originator of sort of the microdialysis movement, studied this in TBI and has used it quite extensively. It's now part of a multimodality monitoring array. And more recently, people have focused on things like autoregulation, and these will be future multimodality markers, NIRS, TCD, and other technology used to identify autoregulation and optimal maps. So what really happened with Lycox? And I have to tell you, I think we kind of put the cart before the horse. So we were looking for a disease-modifying biomarker that we could detect, and based on that detection, change the course of illness. Now, we can talk about the BOOST trial. We can go in and we can say that there is some indication that Lycox, after 10 to 15 years of being out there in the marketplace, has made an impact in TBI, but I can't say that it's a big impact. I can't say that it ever really changes the course of a patient's outcome. It is useful in the sense that when I take care of these patients, it gives me insight into what may be going on, but it hasn't really changed much of anything. Often when I deal with Lycox and multimodality monitoring, I sort of feel like I'm sitting here. I'm the musician on the deck of the Titanic, kind of watching the ship go down. I see all kinds of changes. I try to make interventions. They may have temporary effect, but eventually the outcome is exactly the same. So, what is the medicine initiative right now? The NHLBI has created this initiative in precision medicine. And they've been involved in something called whole genome sequencing in NHLBI, Omics for Precision Medicine, or TopMed. And it's a TopMed program. And what they do is they've collected and assembled omics, RNA-seq, methylation, metabolomics, epigenomics, and proteomics, data within a whole genome sequencing paradigm. Taking all these studies that have all these DNA from outside our community, and they've rejiggered it, so to speak, and they've offered free sequencing while allowing the initial studies to maintain control of the data to some degree, and then to do the analysis. And they want to stimulate approaches that help organize data, ensure they are accessible and interpretable with health disease research, promote discoveries about the fundamental mechanisms that underlying all kinds of population-based disorders, in particular complex genetic disorders, those disorders with more than one gene that influence them. So what about biomarkers in brain injury? So myocardial infarction is sort of the model. The research into this really started back in the 60s. And, one of the things you want when you have a biomarker is you want to look at the specificity of the marker. You want to see it's well-tested and understood biological model. So if we look at biomarkers for myocardium, we had a really great and well-tested model that involved a single organ system with a knowledge about the biological process of ischemia and coronary artery disease. Really, that's kind of the basis of why this was successful markers, because they actually had a biological marker. They actually had an understanding of the disease. And so in the 1960s, they saw the association between things like AST and LDH and CK, and they used this kind of as an indicator and to help them make the diagnosis of myocardial infarction. Then CKB came out because that was a specific CK to heart muscle. And then finally, troponin's INT came out back in the 90s, and it led to the present state. So before you can understand biomarkers and their ability to prognosticate and identify disease, you really have to understand the receiver operator characteristics. So this, again, is something for an example of what an ROC curve is. So over here on the left side, you have the true positive rate. At the bottom, you have the false positive rate. And down the middle, if you had a marker, a blood product, something that would help you to determine whether that disease occurred, if it was ineffective, it would be 0. So if you were below 0, it would actually tell you that you didn't have the problem. If it was above 0, it would tell you with great satisfaction that you had above. And over here, you can see that there's a random classifier. There's better, and then there's worse down here. So what does this translate into in things that we understand and know? So this is an ROC from an article from the American Heart Association, and it looks at sensitivity and 1-my specificity, which is just those same axis, the true positive versus false positive, right? And this is for actually troponin and setting of renal failure. And as you can see, that it actually is probably a better biomarker than one might imagine. Many people are afraid when they look at the troponin levels in the renal and say that if they're elevated, it's just automatically an artifact. But this demonstrates an ROC that's quite high, 0.08, 0.04. And again, when we get into that characteristic of an ROC greater than 0.08, it's kind of the cutoff for which you have an effective biomarker, and it kind of moves its way into the diagnostic paradigm of disease. So remember, when we're using a biomarker like troponin or any other biomarker for disease, say we had one for acute stroke, which really we don't have at this time, is we'd want it to say that disease exists or does not exist. So the variable becomes a phenotype that's discrete. It's either or not. And then the ROC represents that probability that your test is going to be indicative of the fact that disease does occur. So we have arbitrary cutoffs. It's really a spectrum just like anything else. You can have an ROC of 0.6. It just wouldn't be very good. An ROC of 0.8 is pretty good. It means that in general, when you see this elevation, that the patient is in disease. So what are other aspects of biomarkers? So in brain injury, we really lack good biological markers, biological models. We haven't put the basic science effort into understanding what happens. And there's not to say that people haven't done basic science, but they haven't. The brain is very complex. It's not a single muscle system like the heart. There's all kinds of different subsystems. There's all kinds of different impacts of different areas of injury. And then the phenotype is quite complex because it deals with human cognition and human behavior. So when we talk about systems biology and biological model, it's a revolution that's occurred in genetics. So we take genetics, transcriptomics, proteomics, and metabolomics, give us key in association studies to determine what are the underlying biological systems involved in a disease state. So there's kind of two axes to this. So you can get markers from large-scale organization like the entire animal or entire human being, which would be a symptom. Then you can have functional molecules that come from individual organ systems. Then you start to look at regulatory motifs that would work on a cellular level, and then get down into the cell itself, and you can see that there's this progression from genes to RNA expression to proteins to metabolites. So what's unique about this paradigm is you can do association studies across all these and get markers from all across these different parts of the pyramid. Then if you couple them with things like mRNA, genes, proteins, and metabolites measured at once, you can demonstrate a chain of causality from gene through mRNA through proteins. You can find a point and a location at which certain biological systems seem to be playing a role in the progression of disease. This is incredibly valuable. For the first time, we have in our graphs the ability to go and do reverse translational work where we go out and do epidemiologic studies, we collect biological specimens, and we work backwards to understand what biological systems are involved. So the reason we can do this is that there's a discrete bounded data set of genes, roughly 30,000 genes. If we operate on that premise when we do DNA research, we can say not just that we're looking for a needle in a haystack and a possible gene, but we know that there are 30,000 candidates, and we're looking for one candidate that may be activated. And this candidate has association with different biological systems. So that's where we start. We start with genomic data, and then we work across the spectrum to where we look for gene expression in the tissue of choice. Most often, this is actually done in the blood because that's the only organ system we actually have access to where we can readily sample it. Then we also can look at protein expression, particularly in the blood, and then we can look at metabolome in the blood and urine. So what do we know about biomarkers and brain injury? We know the organ system. It's a biological compartment in the brain. We can look for things that have very specific tissue orientation. We want to know something about the half-life of the marker, and is it available by blood test or CSF? So here's a list of all the known biomarkers of significance. I don't want to go through all of them, but I'll just say S100B, normal specific enolase, GFA, tau. These are all very common biomarkers. But there's a whole bunch of other ones that are often associated with specific tissue types, in particular macrophages, and can be very useful. So I took this out of an article that I got from PubMed, and it's from Neuroscience Behavior. They have a nice cartoon. And this is an example of some of the more common markers. So tau, which is related to the microtubule and axons and has some toxic fragments and tau aggregation, is sort of a marker of ongoing axonal damage. GFA, or glial acid protein, is associated with activated microastrocytes and indicates a damage or repair. SB100 is associated with astrogliosis. It really has kind of two components. One is a neuronal component. The other one is an astrocyte component. And together, it seems to have a relatively higher true positive rate for neurologic disease than most other ones. It's most commonly used. It's been used in a lot of different diseases, including trauma, as well as sepsis. And then this is another cartoon, which I think is very important because it shows how these different markers can be accessed. And for instance, this looks at the lymphatic system, demonstrates how it can end up in the CSF or in the serum. And then down here we have a classic model of how these proteins can extravasate into the blood through a disrupted blood-brain barrier junction. Another biomarker that I've worked with is a ceramide, and it's a known potent signaling molecule that mediates key events of cellular pathophysiology, including cell development, growth, and apoptosis. It is a precursor of all complex phingolipids. It's highly specific to fatty-rich white and gray matter of the brain. And its elevated ceramides are seen in C20, C22, C22-1, C24. These are kind of the common fragments. You have to do this. It's a lipid, so you have to do a special lipid test, HPL with lipids, high-pressure liquid chromatography with lipids. And here's the pathway. So it's also known that ceramides are a signaling pathway within cells. An alteration of the ceramide can actually be protective against apoptosis in certain circumstances. It's been implicated in cardiac disease. They're highly enriched in the central nervous system. So no other part of the body has a higher concentration of ceramides. So they're somewhat specific, just as troponins might be specific to the heart. And they're found particularly within the white matter tracts. They're implicated in the development and progression of neurodegenerative disease, such as Alzheimer's disease. We've looked at it in subcortical white matter injury in cognitively normally elderly adults, and it's got utility as an additional marker for white matter injury in small vessel cerebrovascular disease. I'll talk a little bit about that. That's some of our work. So here's a paper that we came out with. So this map here is an MRI, looks at the fractional anisotropy, and it looks for disrupted cortical tracts in white matter-rich regions. We did this across a region, and then we calculated an FA in the tract regions. We found that higher FA was associated with higher levels of sphingomyelin. So if you had disrupted tracts, you had higher levels of ceramide, particularly ceramide 18. And it was really very specific for the genu of the corpus callosum and fornix in our population. Again, it was an association study with a kind of a small number, so it's has not been well verified on a large scale. So, ceramide was really a mechanism-specific biomarker. How would you go about discovering biomarkers? So back in the early 2000s through 2010, there's a gentleman named Lance Leota at the NIH. He discovered a way of finding sort of the needle in the haystack of serological biomarkers and reported some interesting findings in prostate cancer, which initially were exciting but didn't pan out. But what was exciting was the technology he developed. And so he used something called CELD, or surface enhanced laser desorption ionization. And he also used MALDI, which is the same thing, except instead of putting on the surface of a chip, you embed it in a matrix. And so what it does is use a laser to fire away and ionizes these different molecules. It takes time for them to spread across an ionic gradient, and then you can see the rate of speed across that ionic gradient. It also uses MR spect to look at the size of the molecule and some of the compositions of those outliers. So it combines a whole bunch of different modalities, and it was a way of finding very distinct signatures from proteins in the blood. The thing about the blood is really very rich in albumin and some other factors, associated with coagulation, but those are even much less concentrated in albumin. But there's also a whole melu. Potentially all of the 30,000 proteins that are expressed in our human genome could exist inside the blood, but at a very low concentration. So using this technique, you can look at low concentrations and you could still detect a signal comparable to high concentrations. So we did this in a small group of patients for whom we had established that they had aneurysmal subarachnoid hemorrhage and then vasospasm, now DCI. And we discovered a signal. And once we discovered that signal, we categorized what kind of protein it could be. And then this was our CELD profile. We came out with a heat map about all these different signatures. Once we identified the signature, sometimes depending on the state of discovery for that signature, can be related to a specific protein, but not always. Then we went on and used a RATEC cytokine array because we found some signals that we thought were interesting. In particular, we found that ApoA1 and ApoE were serum protein concentrations that were higher in patients who had vasospasm as compared to those who didn't. And we all know that ApoA and ApoE are related in vasculopathies and put people at risk for intracranial hemorrhage, amyloid angiopathy, as well as associations with dementia and small vessel disease. We also found that serum amyloid P and serum amyloid A appeared to be elevated in a small cohort. And amyloid, which is the protein usually associated with dementia, in this case, is also a chaperonin, and it represents injury to cell bodies in general and also in neurons. And so we thought that this extravasation may be related to a cellular injury and a marker of cellular injury. We never really expanded on this. We never were funded to go further after the study. So another protein that I've looked quite a bit at is haptoglobin. It's a complex tetramer glycoprotein. It consists of 2 alpha and 2 beta chains. It's synthesized mainly by the liver. It is an acute phase protein that binds free extracellular hemoglobin after lysis RBCs in the blood and other tissues. It also has other issues. It's kind of a key protein in the blood, and then it has multiple functional modalities, and its control is highly regulated by a variety of different genes. And so this protein probably has a big impact on response to injury, inflammation, as well as the binding of free heme. And it's been a predictor of clinical outcomes in cerebral vascular disorders. And we wanted to determine if the HP genotype was predictive of secondary sequelae and outcomes in ICH and aneurysmal subarachnoid hemorrhage. Again, what's interesting about this protein, this is a complex cartoon. It's from one of the articles that I helped publish that was in Nature. And basically, what's interesting about it is that it has multiple configurations. In the normal state, it's really a dimer, but it can be a trimer or even a pentamer. And this is regulated by a series of mutations that have occurred in the structural portion of the protein. Now, these mutations have not occurred just once, but they've occurred multiple times over the last 5 million years of hominid evolution. And it suggests that this is a very important protein and that these variations, which have reemerged and disappeared, probably have a big impact on survival in certain circumstances. So we did a number of papers. The first one, we looked at worst functional outcome after spontaneous ICH. This really isn't our best work. It was a small sample size. And I'm not sure if it's completely correct, but we went ahead and looked at it anyways. And our biological model was that haptoglobin 2,2 has increased activation of the inflammatory system. And as a result, would probably affect cerebral edema with greater greater outcome or greater impact. Now, the reason I say this first one probably isn't correct is we went on to do another one where, our initial biological model was about cerebral edema. The small study suggested there was a change in outcome associated with IVH, but it was pretty small, very much subject to type 1 error. Then we went and did a larger study specifically looking at changes in edema. And instead of having greater edema with the 2-2 genotype and phenotype, we had less edema and greater edema with 1-1, but it was at the beginning of the process. And we didn't really find a correlation between survival as we had in the other one, but it suggested that the one subtype was much more strongly associated with the early onset of cerebral edema. Now, we can create all kinds of biological mechanisms to presuppose why that might be. The 2-2 may have a greater activation of macrophages, but the 1-1 has a greater affinity, more rapid affinity for binding to free heme, and it can actually then bind to macrophages. So early on, when it sweeps in and sucks up all the dangerous metabolites, it may activate macrophages at a greater rate and result in greater movement of the heme molecule out of the brain. We also looked at this in subarachnoid hemorrhage, and we looked at cerebral salt wasting, and which is kind of a rare phenotype. It was very difficult to find patients with this, and we found a pretty strong association between the 2-2 subtype and cerebral salt wasting. And for those who don't know, when you have subarachnoid hemorrhage, you have this extreme increase in urinary output associated around days 3 to days 21 that is associated with increased mortality. We also looked at the haptoglobin genotype in subarachnoid hemorrhage. So there was a paper that came out by Brophy et al. Many years ago from Henry Ford, and what they found is that the 2-2 subtype was associated with increasing vasospasm and probable increasing mortality. Again, this was not the greatest study in the world. It was a single center study. It was there was no randomization. It was an observational study. The numbers were low when it was subject to type 1 error. So, having said that, it still was a noble effort by the people who looked at it. I think that they had a great biological model. They just didn't have the tools to complete the necessary study. So we wanted to overcome this by looking at people who had looked at this particular issue over the years, including our samples. And this was led by Ben Gastra and Ian Galea from Southampton in the UK. We came up with a consortium called the Hatch Consortium, and we took all the individual patient level data and we created analysis, and we didn't really find an association between the subtypes of haptoglobin and outcomes of subarachnoid hemorrhage. So I think this tells us a number of things. First of all, it doesn't say that there isn't a worse outcome with 2-2. It says that at our present level of power, with our present paradigm of study, we couldn't detect the signal. So, it's not saying that haptoglobin isn't playing a role. It's telling us if it is playing a role, that it's probably smaller than we had anticipated. And the only way to really address this is a much larger study. So even though we had a fair number of patients in this study, it probably wasn't enough and it wasn't designed in the way that allows us to detect the signal. And this is often the case in retrospective studies, even retrospective studies of prospective data. So we published this in Neurology as a negative study. Finally, I'm interested in a new biomarker, and I think that disruption of the blood-brain barrier is one of the first steps that leads to damage to the neurovascular unit. So we came up with a paper, and we're trying to do some more studies looking at mean transit time and blood-brain barrier disruption in patients with asymptomatic vascular white matter lesions. So there's something called the Platak method, where you give gadolinium and it moves across the blood-brain barrier. You measure once before as you give it, and then you measure later to see how much actually made across the blood-brain barrier. They also use this in cancer therapy to look at blood-brain barrier. However, in asymptomatic healthy people, the signal is much less as compared to those patients with cancer. We also used a new methodology kind of put forward by Rich Lee and Peter Van Zoll and Dr. Barker at the Johns Hopkins, that looks at the transition of gadolinium on T1 images in a very rapid sequence. And we kind of put the two together, and we found that we could actually detect blood-brain variability in these patients with a certain amount of sensitivity and specificity. And we're hoping to use this method to look at a variety of different brain injuries, including sepsis, ARDS, and possibly even things like subarachnoid hemorrhage.